السلام عليكم. طيب إن شاء الله المحاضرة دي بالنسبة للجزء بتاعي هتكون المحاضرة الأخيرة. أما باقي المحاضرات اللي هي عن الفاكسينز والمونوكلونال أنتي بوديز إن شاء الله هيديكم لها دكتور علي. إن شاء الله هيخلصها قبل ما تبدو اللعبة. طيب في محاضرة عن البوليمريز تشين رياكشن كان اتكلمنا عن البي سي ار في محاضرة الابليكيشن ان فيترو ابليكيشن اوف دي ان اي ريبليكيشن، فهي نفس الكلام. انا بس نزلت لكم السلايدات لكن ما في شيء جديد، بس اقروا السلايدات. اما بالنسبة للسيل سايكل فانا شايف انه ما في، يعني كلها بالنسبة لكم معلومات المفروض تكونوا عارفينها، فما بتحتاج نضيع فيها وقت. فبرضه نزلت لكم لعبة 3D ماتيريالز، فمروا عليها يعني كده بس، يعني مرور سريع، لان هي البيزكس كلها بتاعت السيل ديفيجن وكده. فإننا اتطرقنا لعملية السيل ديفيجن وكده، اتكلمنا عنها كتير. فإذا يعني استرجعوا الريدي ماتيريال إن شاء الله. بالنسبة للتوبيك بتاع السيل سايكل. طيب بالنسبة لمحاضرة الليلة هنتكلم عن الستيم سيل ثيرابي. هنعرف عن شنو ستيم سيل وكيف ممكن تستخدم في treat certain diseases. stem cell هي يشار لها بأنها هي الماستر cells or the raw material because from which all the body mature and all the cell are differentiated from stem cells. يعني شنو cell differentiation? Cell differentiation is the change of the cell from type to another. So changing the cell from one type to another is called differentiation. أما بالنسبة للstem cell فهي تعتبر undifferentiated، فهي الأوريجين of all the cells. لكنها undifferentiated. إذا حصل لها differentiation السيل بتكون specialized cell، يعني يكون عندها function معينة. أما بالنسبة للstem cell هي فقط تتحول أو منها بيحصل differentiation to other cells، فهي undifferentiated and specialized cell. طيب example: the change from simple zygote cell into complex system tissue or cell type. يعني a differentiation of the zygote or the change of type from zygote cell into more complex tissue system or cell type called differentiation also. طيب من الstem cell ممكن we can get from stem cell brain cells, nerve cells, heart cells and pancreatic cells. So most of the diseases that still there is no cure for it is related to brain, nerves and hearts and pancreas and others, but mostly and plus also cancer. So in this lecture we are going to talk or give an examples of some diseases that they're using or investigating the stem cell as a therapeutic option for these diseases, but not all of them. لأنه إذا تطرقنا على كل الأمثلة المحاضرة ما هتخلص. فهي في الأخير هندي بس examples في الأخير، وفي النهاية الفكرة واحدة. طب نجي لل properties of stem cell. هي زي ما قلنا undifferentiated and also unspecialized cell. طيب أنا في فيديو أفضل إن نشوفه أول، أعتقد بيقرب الفكرة أكتر. The nature of stem cell. بعدين إن شاء الله بسيب لكم اللينك للناس اللي عايزين يرجعوا له. طيب، زي ما قلت دايما الفيديوز دي بيكون بسيب لكم اللينك يا في اليونيفرسيتي بلاتفورم او ويب سايت، او بيكون اذا عندي منه نسخه بعمل لكم له داونلود، او بسيب لكم بعمل لكم له داونلود في الويب سايت، لانه لاحظت بعد المحاضرات بيكون الصوت ما واضح فيها، فاللي عايز يرجع ممكن يرجع لها هناك لانها بتسهل عمليه استيعاب الموضوع كده. طيب زي ما قلنا وزي ما شفنا في الفيديو انه الستيم سيل properties دايما بتكون undifferentiated و unspecialized cell. They have the potential to be differentiated to major specialized cell types or organ in our body, and also they have self-renewing feature. They can self-generate new stem cells. So from stem cell they can produce another stem cell, so they have self-renewing feature. So that is also promising for repairing tissue. So it's a promising feature for repairing tissue, especially neurons. So بعدين هنشوف كيف ممكن the stem cell is now under research for treating diseases like Alzheimer's and Parkinson's disease. So they have the potential to replace cell tissue damage by severe illness. So زي ما هنشوف هنا stem cell could go two directions. Phase one is for self-renewing to give another stem cell and limited cell division. So the second way is to differentiate to specialized cell, for example white blood cells. The classification of stem cells, it could be pluripotent, totipotent, multipotent, oligopotent, and unipotent. For the pluripotent, is this type of cell can be differentiated into almost all cell types, and totipotent also most of the types can be differentiated from the totipotent, and these two mainly obtained from embryos. And for the multipotent, it's mainly obtained from adult cell. It can differentiate into a closely related family of cells. Only for the same family it can be differentiated to another cells from the same family, and oligopotent can differentiate into few different cell types. But for unipotent, it can only differentiate into their own type of cells. طيب the sources of stem cells زي ما قلنا في spare embryo, special purpose embryos, cloned embryos, aborted fetuses, umbilical cords, adults, tissue or organs, or even cadavers. These all sources from which we can get stem cells. The difference between embryonic stem cell and adult stem cell, زي ما قلنا from the pluripotent can be obtained from embryonic stem cells. This stem cell class can become any cell types present in our body. This is one of this that advantage from the stem cells coming from embryo source, and also they are stable and can undergo many cell division. That's mean they have the capacity to self-renew and easy to obtain, but we have to destroy the blastocyst. So in order to obtain these stem cells, we have to destroy the blastocyst. On the other side, for the adult stem cells, it's multipotent class, can become many but not any type of cells. Example, blood stem cells can give several blood cell types. So from blood stem cells it can differentiate to several blood cells. So it's from the same family, but cannot develop brain cells or kidney or liver cells. So this is one of its disadvantage, and also it's less stable and don't have the ability to self-renew, and difficult to isolate from adult tissue also. So here this diagram show from which stages we can isolate the stem cell. So after filtration we got the zygote cell. So from zygote we got morular cells. So from these two cells we can obtain the totipotent cell, and also after that we get the blastocyst and the more complex blastocyst. So from these two we can get the pluripotent stem cell. So these two are embryonic stem cells. So then from it we can isolate it and genetically engineer it in the lab, and then give it back to the patients at the therapy type. So how the therapy work? So when a stem cell transplanted into the body and arrive into the injured part, for example the brain, then the stem cell will come in contact with the gross chemicals and in the body, and these chemicals will program the stem cell and differentiate into tissue surrounding it. So the idea is that we are introducing the stem cell that have the ability to differentiate or to change type into other specialized type of cells. So when we introduce the stem cell to specific location where we require the stem cell to differentiate to give the required type of cell to treat the disease, then in that area, in that location, there will be some chemicals that will give a signal to the stem cell to differentiate or to change its type to similar or to a cell that's similar to the cells in that area or in that location. So for example in the brain, if we introduce the stem cell to the brain, then the chemicals for example growth factors like epidermal growth factor, it will give the signal to the stem cell to be differentiated into brain stem cells. So then how the therapy can be introduced to our body? There are two ways. The first way is direct delivery, and the other way is cell-based delivery. For this direct delivery, so the stem cell will be genetically engineered in the lab, and then will be inserted into a host like retrovirus, and then given to the patient and allow to be differentiated inside the body. The other way is the cell will be also genetically engineered in the lab, but it will be also start differentiation in the lab outside the body, then given to the body as a differentiated cell. Why? To reduce rejection, because the body could reject the stem cell. So by two ways we can minimize the rejection. First, by genetically engineering the stem cell. Second, by also if the differentiation is start outside the body, also it could also reduce the rejection by the body. So this is the two ways or two methods how the stem cell can be delivered to our body. The first example is for the treatment of leukemia. So for the leukemia, we need to remove all the abnormal blood cells or white blood cells. Then we allow the bone marrow to reproduce normal cells. If it's not possible, then we have to start the chemotherapy or even bone marrow transplant. So if we go for the bone marrow transplant, then we need stem cells. So we can get stem cells from healthy donor, then we replace the patient bone marrow cells with the stem cells. Then the stem cells, when we transplant the stem cell in the bone marrow, then eventually it will get the signals to be differentiated into bone marrow cells. Then the bone marrow hopefully will produce the normal leukocytes. There's steps for this kind of therapy. First, we need to reduce the cancer by using the chemotherapy, reducing the number of cancer cells. So then we need to collect the stem cells from the donor, and then there will be a high dose treatment to remove any remaining cancer cells if exist. Then the stem cells will be given by transfusion to the patient. The other kind of therapy is for diabetes. Then the aim of this therapy to make the body produce insulin naturally again and stop the insulin injections. So in a trial or in a study that being conducted for 11 young patients, type 1 diabetic patients, so they were given first immunosuppressive medicine to reduce the chances of rejection for the stem cells. Then they were given stem cells collected from their own blood, and then they give it back to them. Then after that, they have found that actually the patients start to produce their own insulin naturally again, and they're not depending on the insulin injections. So this was research, so only on 11 patients. So it looks also promising, so but still it's under research. Most of the stem cell therapy is under research, but there's some therapy is already approved and used for treating some diseases. The next one is for the heart disease, stem cell therapy for heart diseases. So the aim here to to introduce the stem cell in order to create new blood vessels and to improve the blood flow and generate new tissues and heart muscles. So also the stem cells could be collected from patients' blood and then give it back to the patient, and in order to generate or produce cells that able to create new blood vessels and improve the blood flow in the heart. Okay, then here treatment for nervous system, mainly for the cases of CNS disorders and spinal cord injuries. So in this case, the stem cell could be collected from patient bone marrow and implanted back into their body after few days. So the few days is required for the cell to be genetically engineered and processed in the lab, then give it back to the patient. So this genetically engineered cell, it should be able to regenerate the damaged tissue in the CNS. For example, for Alzheimer's disease, so the stem cell, it will be prepared in the lab, processed in the lab, and then given to the patient to produce neurons to improve the quality of life, improve the memory and behavior by regenerating the neurons. Or for the Parkinson's disease, the nerve cells is unable to produce the dopamine anymore, so we implant the stem cells, then the nerve cells will be able to produce dopamine again, then dopamine will transmit nerve signals. So this is just an example about how stem cell therapy works, and there's so many other examples. إذا واحد عايز يعرف أكتر في examples كتير جدا ممكن تلاقوها، لكن أنا شايف إحنا ممكن نكتفي بالقدر ده في الأخير. زي ما قلنا الفكرة واحدة، والسلام عليكم.